Hello everyone, I am Kamal. I am here to present about our group assignment of the MDA511. The group assignment is about the data set analysis of the diabetes, and in this group we are four members, me along with Ambik, Arusa and Sabina. Here we are going to present our summary in this presentation about what we have done in the assignment and what type of testing we have conducted. In this presentation we are going to discuss about the 10 different topics: introduction to our subject main MD511, and there will be assignment overview, what sort of activities we are performing or we are doing in this assignment. And after that we are going to start with from the content of our dataset, which is the diabetes. First of all, we are going to understand what diabetes is and then the global impact of diabetes. Then we will be discussing about the data selection from where we have selected the data, and there will be like the data preparation, like in which we are neglecting the null values and summarizing the data. After that we are going to visualize the data using the different plots and diagrams like histogram, box whisper, 8 maps. After that we will be discussing about some research questions that are associated with this dataset, and then we are creating some hypothesis, like there will be something as about our perceptions, which will be testing, and there will be a final conclusion. Now let me start this presentation with the basic introduction to the subject. Basically, the MDA511 is the course of the Melbourne Institute of Technology for helping the student to equip with statistical data necessary for data analytics. It is basically about the linear algebra, description, statistic, probability, elimination, hypothesis testing, etc. And we don't need prior knowledge of like programming or coding for the data analytics to overcome this subject. This subject is like a path showing for the students who are just starting for the data analytics program. Now let's over go with the assignment. This assignment is about the statistical explanation of diabetes dataset. The main goal is to identify, analyze the prevalence and risk factor associated with the database. The tasks aim to pinpoint potential predictors for the disease using a range of statistical tools like I have previously mentioned, like correlations and showing how different factors are interrelated with each other for causing the diabetes. And after going through this assignment overview, let's go on like understanding what the actual diabetes is. Diabetes is the chronic health condition affecting the how the body turns the foods into energy. Basically we can say that we consume different foods that are basically converted into the in the form of the energy, and the malfunctions or unable to generate certain energies, we can say as diabetes. It involves the hormone insulin produced by pancreas, which helps to regulate the blood sugar level. Now, after studying this case, we have researched some of the sites like the WHO and the Australian health regulation, from which we got to know that in 2022, approximately one in every one in 20 every Australian citizens have the diabetes, which is roughly around 5.3% of the total population. And talking about the old, there are 443 million people who have basically the diabetes. Now, my friend Ambik will continue this presentation from here. Thank you.
Thank you, Kamal. Now I'm taking these presentations. Hello, my name is Ambikale. Now I'm going to talk about global impact of diabetes. Globally, about 422 million people have diabetes, with a majority living in low and middle income countries. The increasing prevalence of diabetes poses a significant public health challenges worldwide. Effective management and preventions require as a comprehensive, understanding of risk factors, and treatment approaches. The highlights and the importance of diabetes and research to combat the growing diabetes epidemics. Now, data selection. The diabetes data sets are collected exclusively from Kaggle, ensuring a reliable data source for analysis. Kaggle datasets are renowned for their quality and provide a foundation for strong statistical analysis. Data selection is a critical step impacting the accuracy and relevance of the study findings. Using a trusted source like Kaggle enhances the credibility of research and its outcomes.
I'll be talking about data preparation and data visualization. For the analysis of dataset, we need to prepare our data. The first step is to load the dataset into our Python notebook. To check if the data is loaded properly into our notebook, we use head and tail to print the first 5 and last 5 data respectively. And then we check the number of columns and rows and the type of data used in the dataset. The next step is to check if there are any null values or if any rows are duplicated. We replace the null values with the aggregate of the total value of the same column. To know more about the dataset, we use describe. It gives us the count, mean, standard deviation, minimum and maximum value. Lastly, we copied our dataset to a new variable to compare the raw data with the clean data. For data visualization, we used scatterplot, histograms, and heat maps.
After the data selection and the data preparation, the most important step is the data visualization. The data visualization is the major part since it affects the analytics, and if we use the graphical representation, it is very easy to understand and analyze the results and the outcomes. For this presentation and for this assignment, we have used various graphical representations like histograms, box plots, scatter plots, and the heat maps. Histograms and scatter plots reveal patterns and distribution within the diabetes dataset, whereas heat maps and box plots provide deeper insights into data relationships and outliers. For example, we have this histogram where we can see the frequency of the pregnancy and the glucose. As per the histogram, the frequency of the pregnancy is around 250 and similar to the glucose. If we see the blood pressure and the insulin, the frequency is about 200 more than 250. For insulin, it is more than 400, which reaches up to 500. If we see the outcome, 0 here, 0 means the people without the diabetes, whereas 1 means the people with diabetes. People suffering from diabetes reaches around 500, whereas the people with diabetes is around 250 or 300. So seeing this outcome, the population sample size we can estimate is around 800 people only. If we see the scatter plot around about this, we can see like the linear positive correlation as it follows the linear shape, linear single line. If we see the correlation between the glucose and insulin, it shows the positive linear correlation as it follows a single linear line. It says that if the glucose level increases, the insulin also increases. Similarly, if we see the scatter plot of skin thickness and the BMI, which is body mass index, it also follows a single positive linear regression, which says that there is a correlation between skin thickness and BMI. Hence, the data visualization is very simple to understand if we understand the regression line about this linear positive correlation. The next one is the heat map. Heat map is one of the simple thing to understand as it gives the numerical value and the correlation values. By using the color indication, which says greener the side, more correlation is there. Which is if the number is towards the green side, there is a more correlation values. If you see the pregnancy, the more green side is 0.54, which is related to age. Hence, it proves that pregnancies and age are correlated. If we see the glucose, the more greener side is 0.47, which is related to outcome. Outcome means those which means that people with diabetes and people without diabetes. Hence, the heat map is very useful data visualization to understand whether there is a correlation between two factors or not. Next, we have the correlation numerical values, which gives the correlation between two factors. If we see the most correlation values is maximum is 0.49, which is glucose. Hence, it is clear that the glucose has the maximum correlation with the outcome, followed by the 0.23, which is the age. This means glucose and age are the top most affecting factor for the diabetes. So this data visualization and this analysis solves the research questions and also creates few research question which needs to be solved. So the research questions are very important for the next generation, whether the questions are solved or unsolved. Clear research questions are essential for a focused and effective statistical examination, since research question is the single reason that we use for the data analysis and for the interpretation. This presentation is based about few research questions, which are what are the risk factors of the diabetes. As we have discussed here about various factors like age, pregnancy, BMI, skin thickness, what are the most risk factors of the diabetes? The second question is, are the age and diabetes are correlated? And the final question is how the lifestyle factors such as BMI, body mass index, and the obesity of the body affect the prevalence of diabetes. These research questions are solved by the hypothesis testing and their result. Hypothesis testing is about the statistical analysis, which gives the p-value, and according to the p-value, we describe whether it accepts the null hypothesis or rejects the null hypothesis. For the alternate hypothesis, we have two types, which is one-tailed and two-tailed. One-tailed means it only goes in one direction, such as either it increase, either it decrease. The one factor is increasing or decreasing the other factor. For example, here, if the age is decreasing the prevalence of diabetes or decreasing the prevalence of diabetes is the one-tailed factor. While two-tailed testing or alternate hypothesis is, it goes with two dimension, which means either it affect or doesn't affect the diabetes. For this presentation, we have tested three hypothesis testing. The first test is between glucose and diabetes. For this, the null hypothesis is that the level of glucose is not correlated with the likeliness of diabetes, and the alternate hypothesis for this is the level of glucose is correlated with the likeliness of diabetes. If we see the p-value of this, which is 2.90, if the p-value is less than 0.05, then only we can accept the alternate hypothesis. But the p-value here is 2.9, which is more than 0.05. Thus, it accepts the null hypothesis and proves that the level of glucose doesn't affect the diabetes. The second test we conducted is about age and diabetes, which is our second research question, whether age and diabetes are correlated or not. The null hypothesis is that age is not correlated with the likeliness. In the contrary, alternate hypothesis is that age is correlated with the likeliness of diabetes. Here also we can see the p-value is more than 0.05, which is 2.2, and it also accepts the null hypothesis. Same result we get in the test 3, which is conducted for the blood pressure and diabetes. The p-value is 3.7, which is more than 0.05. Thus, it also accepts the null hypothesis. All the 3 tests tested the hypothesis testing gives more than 0.05 p-value, which all accepts the null hypothesis and rejects the alternate hypothesis. These results may vary according to the population size. For this data set, there are only 800 samples, which may vary the results and the analysis. So for conclusion, the sample size of the data set affects the analysis, interpretations, and the result. The data set are collected from the Kaggle website, which is one of the most renowned and accurate data information site. And for the data visualization, we have used various graphical representation like histogram, scatter plot, heat map, etc. And the analysis is done by the hypothesis testing to solve the research question. Thank you.